Breakthrough. Scientists identify new target for Parkinson's therapies. I'm Steve Blair. Welcome to Breakthroughs. A master control region of a protein linked to Parkinson's disease has been identified for the first time. A new finding made by scientists from the University of Leeds provides a new target for the development of therapies to try and slow down or even prevent the disease. Parkinson's affects more than 10 million people across the world, causing neurodegeneration and difficulties with movement, which increase over time. There is currently no cure for the disease. The study focused on a protein called alpha-synuclein, which is linked to the onset and progression of Parkinson's disease. Alpha-synuclein is found in healthy cells in the nervous system, but problems arise when it clumps together or aggregates into plaques known as amyloids that can disrupt normal function. A short region of the alpha-synuclein protein, known as NAC, was assumed to be the key to Parkinson's disease, as it's particularly aggregation prone. In this study, researchers found that two regions outside of NAC played a critical role in controlling amyloid formation in the alpha-synuclein. Removing these regions switched off aggregation in a laboratory setting, even though NAC was still present. They inserted alpha-synuclein and a variant of the protein lacking the master controller regions into the muscle cells of nematode worms and monitored aggregation of the proteins and their effects on the mobility of the worms, a commonly used model organism in research into neurodegenerative disorders. When the controller regions were deleted in the worms, alpha-synuclein no longer formed aggregates, and the worms were healthier and more mobile, even in old age, compared to worms expressing the normal alpha-synuclein protein. Their findings were published today in Nature Structural and Molecular Biology. In trying to tackle diseases like Parkinson's, the first problem is identifying the key areas to target with small molecules or protein-based medicines, as these proteins do not have a fixed structure, ruling out traditional methods of structure-based drug design. Finding a previously overlooked target to focus future efforts on is very exciting. Our discovery of master controller regions may open up new opportunities to understand how mutating the protein sequence that causes disease could help us find the Achilles heels of these proteins to target further therapeutic intervention. Whilst alpha-synuclein is linked to Parkinson's disease, it is also thought to be involved in signaling across nerves in the brain. This involves the release of neurotransmitters which occur when small carrier structures called vesicles containing vital signaling molecules fuse with cellular membranes to release their cargo. This is important for signaling in the nervous system. To investigate whether the master controller regions identified in alpha-synuclein are also important for its function, the researchers looked at whether the modified protein could still fuse vesicles together. They found that while removing the master controller region prevented aggregation, it also prevented vesicle fusion. This suggested a tug-of-war between the function and harm aggregation of the master controller region. Fine-tuning this balance may enable therapeutic opportunity to reduce aggregation whilst maintaining function, and it is the need to rebalance these two opposing roles of the master controller region that will be the next challenge. We hope that such a strategy might be able to help people with early signs of Parkinson's by reducing the formation of amyloid plaques in the brain and to delay the progression of the disease. Well, that's pretty cool. I'll leave a link in the description to the full article, and as usual, please like, share, subscribe, and take care.